and over to you.
Thanks, Mark. It's an absolute pleasure to be here. It's always nice to speak to the UCT crowd. Thanks for the invitation. Mark asked me to talk about hot topics in fungal infections, and that sounded too much like a conference presentation. So I decided that I would just focus on Candida auris. And I think there's sufficient there's sufficient updates in this area to warrant a talk on this. Certainly in South Africa, the epidemiology has shifted dramatically in the last few years. The new diagnostic assays focused on Candida auris, the new antifungal agents with activity against Candida auris, and we've also gotten some new strategies for surveillance, which is really exciting. So Candida auris behaves like a multi-drug resistant bacterium. It's a relatively new pathogen based on molecular clock work, looking at the phylogenetics. It seems to have emerged in the last 300 or 400 years. The drug-resistant strains, which actually caused the large healthcare-associated outbreaks, probably emerged in the last 40 years or so. But really, we only detected Candida auris in a clinical setting in around 2009, coincidentally when we first found Candida auris in South Africa too. It's now globally distributed across 6 continents. And as I said, it really behaves like an MDR bacterium in many ways. Its outbreak potential, the multi-drug resistance, the fact that it's difficult to identify, the recommended antifungals, primarily kind of candins, are not widely available in every country where this pathogen is spreading, and we don't have very good preventive measures, unfortunately. So the WHO issued the first fungal priority pathogen list last year. And as I was saying to Mark, before we started formally, but this really has already had a material impact on global agencies thinking about fungal pathogens, and in particular Candida auris, which was listed as a critical priority pathogen alongside the major human pathogens, Coccus, Aspergillus, and Candida albicans. So for this talk, what I thought I'd do is just use a really simple conceptual model for Candida auris transmission in a healthcare setting. And those of you who've done epidemiology would recognize, a really simple compartmental model. People who are infectious with Candida auris are either infected or maybe colonized, and both infected and colonized people will shed Candida auris into the environment, and also this facilitates transmission to susceptible people in the immediate environment. Contaminated hands, healthcare workers may be a secondary means of transmission. So we'll use that model as I discuss this. I also want to quickly tell you about laboratory-based surveillance for Candida infections and colonization in South Africa, because I'm going to present some of the epidemiology data, and it's useful just to understand how we've collected these data. So we use a network across South Africa called Germs South Africa, a network of public and private hospitals. And it's passive surveillance in the sense that we're essentially allowing clinicians to collect specimens from people with clinical syndromes routinely. And of course, the diagnostic laboratories then isolate, identify, and perform susceptibility testing for Candida. From a surveillance point of view, we then look at culture-confirmed cases, and those cases, the electronic data, and in some instances, the isolates, as well as at Sentinel hospitals, case report forms are then sent on to the NICD. The isolates are being processed at the mycology reference lab where I work. So let's start with susceptible. Who's susceptible to Candida auris? And of course, the risk factors for Candida auris infection or colonization are fairly typical for any Candida species, really. Most transmission occurs in a healthcare setting. People at highest risk, of course, have underlying diseases, and of course, interventions in the healthcare setting, broad spectrum antibiotics, being mechanically ventilated, abdominal surgery, etc. COVID is, interestingly enough, a risk factor for Candida auris, both invasive infection and colonization. And the pandemic obviously created some conditions which facilitated Candida auris transmission. You know, during the peak of some of these waves, there were overwhelmed ICUs, suboptimal IPC, temporary staff came into these facilities. And so really ripe conditions for transmission of pathogens, including Candida auris. And of course, people with severe COVID are vulnerable to bacterial and fungal infections, including to Candida auris. This map just shows you countries which reported COVID-associated Candida auris, either infections or colonization. In South Africa, what we've done is we've superimposed our cases of candidemia against cases of COVID infection and admissions. So essentially, the purple line that you see on the graph is just a 30-day moving average of the cases of candidemia. In 2019, the green bars is essentially just the number of cases, but I think the moving average is easier to see. And then what I've done here, we just superimposed those lines on the the COVID waves. So we obviously had the ancestral Beta, Delta, and Omicron waves. The blue bars showing you the infections, and the yellow the hospital admissions. And I think that you can appreciate that after each of the pandemic waves, there seems to be an increase in Candida auris candidemia. Of course, the ancestral wave also seems to have been preceded by a peak too, and I hypothesized that this may be related to the hard lockdown. So there possibly was, short staffing at that time and more transmission. When one looks specifically at Candida auris and parapsilosis candidemia, because of course those are transmitted in a healthcare setting, I think you can see those, those spikes more clearly post COVID wave, post each of the COVID waves. So it's an interesting phenomenon. Obviously, these are ecological data, so we can't say that this is causal in any way. When we look at the species distribution of the 6 most common causes of candidemia over a 4-year period, 2019 through to 2022, I've highlighted the proportions of Candida auris in red and the proportions of parapsilosis in orange. What you can see is the proportion of auris increases in the private sector with a decline in parapsilosis. And there are also numerical increases in both auris and parapsilosis candidemia, but a much more rapid increase in auris. And you definitely see the peak numbers in 2021. So I believe this may be a replacement of parapsilosis by auris. They occupy roughly the same ecological niche and have the same mode of transmission, but auris has some advantages in terms of its broader resistance to antifungals and disinfectants. So we've seen a similar picture in the public sector. It's not quite as dramatic, though. It seems to be a slower replacement of parapsilosis by auris. Okay, so let's move on to those who are infectious and infected, so usually invasive disease. I want to start just by talking about lab identification of Candida auris, because I know there will be some clinical microbiologists on the line. So, of course, in terms of identification of cultured isolates, we rely preferentially on MALDI-TOF identification and molecular assays, because really the biochemical methods we have are problematic. Vitek, for instance, really doesn't consistently allow identification of Candida auris. I think relatively few labs use the microscan walkaway, but those still produce incorrect identifications because auris, as far as I know, is not on the database. And just remember, the biochemical assays really identifies to species level based on a percentage matched to the assimilation and fermentation pattern. So they'll quite happily give a wrong identification rather than no identification, which can be a problem. Just, for interest, there are some direct detection assays. The BioFire film array panel for blood cultures and the GenMark blood culture ID panel, the FP panel, both will detect Candida auris directly, which is really useful. So in terms of who else, acquires Candida auris candidemia, what's really interesting from our surveillance data is that you can see that, the median time to a positive blood culture from the time of admission is about 4 weeks, roughly. And of course, during this time at risk for Candida auris, these people being admitted are obviously admitted to ICU, mechanically ventilated, have central venous catheters inserted, are started on PPN, are started on antifungals and broad spectrum antibiotics. Just to note that the crude mortality is somewhere between 40 and 60%. We don't have an idea of attributable mortality. This is really, these patients generally are very sick anyway. It's very hard to say what the attributable mortality is. When we first looked at Candida auris candemia in 2016 and 2017, we noted that the vast majority of cases really were concentrated in Gauteng province. Of course, this is where most of the ICU beds really are in the country. But there were relatively few cases elsewhere. Just to note also that we also looked specifically at neonatal cases, and we found very few facilities reporting neonatal cases in 2016 and 2017. Since then, we've continued to look at the data, and in 2019 through to 2022, things have changed. So the vast majority of the cases still remain diagnosed in Gauteng province, but we're seeing more cases in KwaZulu-Natal, the Northwest province, the Eastern Cape, and also the Western Cape. So really spreading across the country. And I think once this pathogen becomes established in a healthcare facility, it becomes very difficult to get rid of it. So just to take you back to some, data about the emergence of this pathogen. So when we first looked at these, at this pathogen, maybe about 10 years ago, looking at whole genome sequence data, the initial data really seemed to suggest that there was simultaneous independent emergence of Candida auris in 4 regions, because at the time there were 4 major clades that we could identify, and these clades are separated by hundreds of thousands of single nucleotide polymorphisms. More recent analyses using a more diverse set of isolates from 19 countries have allowed us to use molecular clock assays to apply molecular clock analyses, And so, it looks as though Candida auris probably emerged in the last 300 or 400 years. Clade 2, which was the original clade first described in Southeast Asia, seems to be the oldest clade. And that's the one that where the name auris comes from. It's got a predilection for ear infections and not so much invasive infections. Clade 4, which is the South American clade, seems to have emerged most recently based on this analysis. And as I mentioned at the very beginning, the drug-resistant strains causing these major outbreaks currently seem to have emerged within the last 40 years. And of course, if you work back to 40 years, that's the 1980s, and that's coincidentally when azoles became available both in human medicine and also used in agriculture. So definitely some human intervention in terms of what's emerged. Just in terms of treatment and what we've got to treat fungal infections, we've got really few systemic antifungal classes, the polyenes, the azoles, the candins. Fortunately, there's some new agents in the pipeline, the GWT1 inhibitors typified by manager picks, the triterpenoids, ibrexafungerp, which is very similar to the echinocandins, and there's some modified agents like resifungin, which is an echinocandin which can be given once weekly, which is really nice. But of course, sometimes trying to get hold of these antifungal agents feels like reaching for the stars. And many of them are actually completely out of reach, particularly in resource-limited settings. So let me just quickly tell you about some of the susceptibility test data that we've got on the Candida auris isolates. But I'm going to preface this with some discussion about antifungal susceptibility testing. So there are no formal breakpoints endorsed by either the Clinical Laboratory Standards Institute or UCAST, the European group. There are tentative breakpoints which were proposed by CDC, but these have not really been correlated with clinical outcomes. So we're not certain what this means. And of course, as with any antimicrobial susceptibility testing, there are always specific issues. I won't go into that unless there are questions. But I'll tell you about some of our susceptibility test results. So between 2018 and 2021, over a 3.5-year period, we asked all laboratories, public and private, to send us suspected and confirmed Candida auris isolates to our reference laboratory for additional testing. You can see that we had roughly the same number of invasive and non-invasive isolates for testing. These were not, obviously representative of all cases, but a fair proportion of cases had viable isolates. For Fluconazole, the tentative breakpoint is 32, and you can see the vast majority of isolates fall to the right-hand side, indicating, resistance, I suppose. And of course, we know that you would never use Fluconazole for treatment of auris. Resistance is mediated by EG11 mutations, and they are specific clade-specific mutations, which is interesting. Amphotericin B, the tentative breakpoint is 2 micrograms per mole. And again, you can see that the vast majority of isolates fall to the left, so would be considered susceptible. There are a few isolates which have MICs which are somewhat elevated, particularly non-invasive isolates. We're not certain what this means. It may be an artifact. There's been no clear documentation of treatment failure with amphotericin B. And then micafungin, the tentative breakpoints for the entire echinocandin class, and micafungin just being the agent you test to work out susceptibility, for example. Most isolates, because most of our isolates are clade 3, are susceptible or within the susceptible range. There are some isolates which would have higher MICs, and again, these seem to be non-invasive isolates. Just in terms of our, the clade distribution in South Africa, so we've looked at about 115 isolates by whole genome sequencing. Most of the isolates are clade 3, shown in the red circles in this phylogenetic tree. Some are clade 1 and 4. A few of these isolates, as I've shown you already, I suppose, are echinocandin resistant, shown by the yellow outer bar, and almost all of them are Fluconazole resistant, shown by the pink outer bar. And then, we've just looked at the point mutations, which are fairly typical for what we would expect. Concerningly, we have found a few isolates which are considered pan-resistant. These have all been found in Gauteng province, all in the private sector, all in a non-ICU setting, and all from urine specimens. Of course, Candids don't penetrate particularly well into the urinary tract. So if someone is colonized with Candida auris or has a UTI, and they're exposed to Candids, there's of course a chance of resistance developing on treatment. And this has been documented in case studies, for example, here, where high-level Candida resistance was documented in a patient. Initially, an isolate which found in the blood and in the urine was a Candida susceptible. I think that was day 27 or so, and then by day 90 something, the patient had a Candida resistant auris isolate on blood culture following exposure. So of course that does occur. Equally concerning is the fact that you definitely see transmission of resistant strains. For instance, in the US, they saw 2 clusters of pan-resistant and Candida-resistant isolates. There were no epidemiological clusters or links, between the 2 clusters. None of these patients had prior Candida exposure, which really suggests transmission within healthcare facilities, which is very concerning. So, how can you treat some of these pan-resistant strains? Ibrexafungerp, as I mentioned, is this novel class, belongs to the novel class, the triterpenoids. It's currently marketed for vulvovaginal candidiasis, but it's got very high tissue penetration into the kidney and bladder. There's a clinical trial currently recruiting in South Africa and India. And incidentally, the company presented a few cases of urinary tract infections at ECMID this year. Patients with auris UTIs treated with ibrexafungerp, and you can see that in this small case series, 3 or 4 patients had a complete response. It's very difficult to treat auris and glabrata infections with our current antifungals, and it's a huge advantage that ibrexafungerp is an oral agent. Managepix is another novel agent. A very small phase 2 study conducted in South Africa among patients with Candida auris candidemia showed very good treatment success. 8 of 9 patients did very well. I don't think that these were patients with pan-resistant strains, though. They were fairly susceptible. And then, of course, just in terms of, where do we go from here in terms of combination antifungals, some really interesting in vitro data from the Exeter group showing that by checkerboard assays, you can definitely find synergy between inidulafungin and flucytosine and inidulafungin and managepix. And this is just illustrated here by these live cell microfluidics imaging pictures. You can see that, for inidulafungin and managepix and for inidulafungin and 5-HC, very few cells remaining. And these combinations are particularly useful for persistent infection, recurrent infections, and also complicated infections. We're seeing, for instance, endocarditis increasingly, which is difficult to manage. So I'm going to move on to those who are infectious and colonized. Of course, colonization is a risk factor for subsequent invasion. It's been estimated that around 10% of colonized people will then develop invasive disease, but there's also a reservoir for onwards transmission, either directly or via shedding into the environment. And so just in terms of, a colonization and decolonization strategy, a search and destroy strategy, which has been used for MRSA, for instance, and to some extent, I suppose, for CREs, but the destroy part in CRE is much, much harder. The question is, could you apply the same strategy to Candida auris, and in which circumstances? So the first thing is, you've got to find Candida auris colonization. And so you've got to decide which part of the body you're going to swab. Candida auris is preferentially seems to be a skin colonizer rather than a gut colonizer. And I'll show you some data shortly to show that the more body sites you swab, the more likely you are to find Candida auris. Generally, we tend to swab the axilla and groin, and if you add in the anterior nares, that increases sensitivity. You can use culture-based methods as well as molecular assays. And just to show you a picture of a new chromogenic agar called Candida Plus, which differentiates Candida auris very well from other Candida species. Candida auris colonies appear light blue with a blue halo. So really is a very useful, quick, rapid assay. There are some false positives. So, and with closely related Candida species. So you've got to confirm this using some other identification method, Multitel or molecular assays. And then just to mention that, molecular assays, direct molecular assays from swabs can also be employed. And obviously there's also the T2 MR assay, which is extremely expensive, but has been used in a colonization detection strategy, even though it's still only research use. So in terms of searching for Candida auris, this was an in-depth investigation in a US skilled nursing facility, which was endemic for Candida auris. They essentially did all these point prevalence surveys over a period of time from 10 body sites to determine the spatial extent of colonization. And they found that the nares were most densely colonized. We added in the axilla and groin, which are commonly selected sites for screening. It takes the yield up to about 80%, but you need to swab many more sites to get 100% sensitivity. And in the same sort of situation in these long-term care facilities where people are ventilated for prolonged periods of time, and there's ongoing transmission of Candida auris. If you do repeated point prevalence surveys, you just see the prevalence increasing over time, likely because of unrecognized colonization on admission and ongoing transmission at the facility. You can see in this particular study, the prevalence plateaued at around 70%, so sort of a steady state of admission and discharge of people colonized with Candida auris. In terms of the colonization dynamics, colonization persists for a really long period of time. Some people have been found to be colonized for longer than 2 years. And what's really worrying is that colonization swabs alternate between detecting and not detecting Candida auris, so they're not that accurate. This is again data from a skilled nursing facility. There were some data from an acute care facility in New York City, where they looked at colonization post-discharge, and they found that the time taken for patients to become serially negative was about 9 months. And the first swab, which was negative, was around 5 months. So once you've left an acute care facility or possibly a long-term care facility, and you're not exposed to ongoing transmission, you'll probably lose Candida auris as a colonizing organism, within a few months, because it's probably outcompeted then by other commensals. In terms of how to destroy Candida auris, there are no clear protocols for decolonization. And of course, the problem is, what's the benefit? What about recolonization in the same environment? One option is chlorhexidine. In these long-term care facilities, they found that you could decolonize residents if you achieved very, very high concentrations of chlorhexidine. But those concentrations were achieved at very few body sites. I think around 10% of skin sites that were tested. So it's very difficult to then achieve the right concentrations for the right amount of time to decolonize. There are other options: povidone, iodine, trimazole, terbinafine have been tested in animal models. What's interesting from mouse models is that Candida auris appears to enter the dermis with no inflammatory response, and so sort of hides away from, it can be undetectable in the dermis and then sort of just reappear. So when I mentioned earlier that colonization swabs can be positive and then negative and positive, it's possible that it's because the pathogen itself is hiding away, and not because the swabs are not sensitive or the assays are not sensitive. I just wanted to show you some data from a large academic hospital in Johannesburg. This was we looked at the situation in the entire hospital, Candida auris cases. These are cases of candidemia over five-year period. You can see that initially there was circulation in the burns unit, mostly in the pediatric and adult burns units, and then a few sporadic cases of neonatal infections, 2017, 2018, and then in 2019 this enormous outbreak emerged in the neonatal unit, which was really very concerning. We've continued to track this particular outbreak over a prolonged period of time, and it's been very, very difficult to get on top of this, despite enhancements to infection prevention and control, attempts at improving antifungal stewardship, attempts at improving environmental cleaning. What we eventually did do in November 2021 is conduct a unit-wide point prevalence survey. Essentially, the unit was planned to move to a new facility at the sort of in mid-November. So we thought we'd use the opportunity to do a point prevalence survey and then give the results back to the clinicians so that they could then isolate cohort patients who were colonized rapidly. We used a molecular assay, we used PCR, and we also used a culture-based assay. The PCR assay was available in about 48 hours, so we gave them those results, and they were then able to cohort patients. Unfortunately, the PCR sensitivity wasn't great. It was only about 45%, so we missed many of the cases. But at least it sort of reduced transmission to some extent. And we have found, by the way, the overall prevalence in that unit was about 33% on the day of that unit-wide survey. But it did seem to have some impact subsequently, of course, confounded by the fact that they moved to a slightly better facility in terms of the layout and infrastructure. Just to talk about the contaminated environment a bit. As I said, people who are colonized or infected with Candida auris will shed Candida auris into the immediate environment, and Candida auris will remain in the environment for ages, for up to 4 weeks, and of course will contaminate equipment and will remain viable on plastic even with cleaning. So it really is a very difficult pathogen to manage. Just to point out, there was a nice outbreak report a couple of years ago from this UK ICU. They had 70 patients with auris colonization or infection. There were only 7 invasive infection cases. These were all clade 3, so the African clade. And all of this all seemed to occur in one particular ICU. The risk of Candida auris infection or colonization was associated with the use of this reusable skin surface axillary temperature probe, which had been used in 86% of the cases. Auris was also isolated from those temperature probes, and the isolates, clinical isolates, and these isolates from the probes were genetically identical. And the outbreak ended once those probes were removed, so it wasn't as straightforward as that. So, in terms of cleaning, Candida auris is resistant to some disinfectants, the quaternary ammonium compounds. As I said, it remains viable on surfaces for up to 4 weeks. You can culture it from bedding for about 7 days. And patients can acquire infections from the environment within 4 hours. These are just data from a UK outbreak. In terms of cleaning, bleach is really the mainstay. You can also use peracetic acid if you add that to bleach to clean plastic. Non-touch methods, so UVC, disinfection, and hydrogen peroxide vapor really should be considered adjunctive, particularly the UVC light, because that really depends on the UV system, the distance to the contaminated surface, the soiling of the surface, the exposure time, the angling of the radiation, the shadowing, and all sorts of other things. Terminal cleaning is also helpful. And then I'm just going to finish by talking about surveillance for Candida auris. We don't really know what the prevalence is in the community, but as more and more people are discharged from healthcare settings, there probably is going to be there will probably be some evidence of Candida auris in the community. Screening on admission to hospital is generally not recommended. In the UK, between 2017 and 2018, they screened about 1,000 admissions to 8 ICUs in 3 cities and found no screens that were positive. In India, though, they screened people with chronic respiratory diseases, and they found that 10% of those people were screened positive on admission. Of course, it's hard to say whether this was community acquisition or it was, just because these people with chronic respiratory disease had multiple healthcare contacts. In terms of surveillance, I've shown you what we've done from a laboratory point of view. The other options are syndromic surveillance, which is obviously much more expensive, and environmental surveillance. So I'll tell you a bit about environmental surveillance because that's really interesting and something new. So some exploratory work, wastewater surveillance for Candida auris. We know that wastewater influent or hospital effluent has been used for CRE detection. And in the US, in Nevada, they've looked at wastewater influent from 2 treatment plants. In Nevada, they really only found their first cases during the COVID pandemic. And by June 2022, they had about 22 healthcare facilities affected by either Candida auris infections or cases of colonization. Most clade 3, which is our African clade. They processed the wastewater from these treatment plants using a selective medium where they added Fluconazole to Savrod agar. And that, of course, screens out, allows only Candida auris to grow. And they found 2 auris isolates from wastewater, which were genetically identical to the clinical isolates in Nevada. The question is this method sensitive enough to detect auris in wastewater, before there's clinical detection in an area? And that's not clear yet. They've taken it one step further in Nevada. They've looked at wastewater now at a community scale in the Las Vegas metro. So over a 10-week period, they're collecting samples from multiple treatment plants. Then they seem to process the pellets, not the supernatant. And this map on the right-hand side shows you what they call the sewer sheds. The color is proportional to the average influent wastewater concentration of auris. And those black diamonds show you the 22 facilities reporting auris cases. There were consistent concentrations over the 10 weeks and a rough correlation with the proportion of facilities reporting auris cases. And this is really interesting because it could allow zooming in. You could then zoom into what they call strategic manholes or buildings or individuals to actually look for Candida auris. They are obviously not issues, but just to say that we're doing much the same thing here. We have set up wastewater surveillance, obviously, for COVID, and we've been processing samples, and we've been able to isolate Candida auris from wastewater samples in culture. I'm just going to end by showing you the satellite photo of Africa at night. So this is sort of an analogy for Candida auris. Of course, in South Africa, the lights go off fairly frequently. But luckily for auris, that's not been the case. When you search for Candida auris and Africa, you find very few articles. A few cases been detected in Nigeria, Kenya, Algeria, but really the rest of the continent, not much else. We've got a new surveillance network that we've set up. We're currently collaborating with 5 countries, Angola, Zimbabwe, Zambia, Mozambique, and Botswana. And we've already found Candida auris, or helped one of the countries confirm Candida auris colonization and infections, which is a big, really useful, because they can actually make a real start at trying to control these infections, as opposed to doing what we did, which was not recognizing that this was going to be a major problem and acting far too late. So just in summary, auris is now the #2 cause of candidemia in South Africa. There's been a huge increase in case number over the last few years, alongside the COVID pandemic. We're seeing new at-risk populations, and we're hoping that this new fungal disease surveillance network in Southern Africa will help us look for or find Candida auris where transmissions are occurring currently undetected, and then work out exactly how we're going to try to control the situation. But you absolutely will find Candida if you look hard enough, and definitely can be found. Thanks. I'm going to end there, just acknowledging the germs network. Thank you.